All right, well, welcome everyone. We'll get started. My name is Maddie Meddy, and I'm a member of Pages Early Career Network. I'm also on the steering committee for the Pages ECN. The North American regional representatives have started this series of show and tell events. They're just this year in order to meet and highlight some of our early career peers and learn a little bit about research and researchers in our region. And these events are meant to take a more behind the scenes look at research and be a little more interactive than what we're typically used to with webinars over the past year. A couple months ago, we had our first event with Dr. Dan Killam at the University of Arizona, showing us his lab setup, growing giant clams at the Biosphere 2. And if you'd like to view that, if you missed it, the recording of it is on YouTube. So you just go to YouTube, type Pages ECN in the search bar, and it's pretty easy to find. Dan was growing clams in the Biosphere ocean in order to better understand their growth habits and further develop their proxy applications. And in today's event, we're going to be looking at clamshells again, but at a much later stage in the research process. And I'll let Nina explain that more fully. So I'll introduce Nina in a moment, but just to let you know, feel free to type questions in the chat box. The moderators will make sure they get answered. If you're comfortable unmuting yourself, you can go ahead and do that at any point and ask a question, but there'll also be plenty of time at the end to do that. And it's great if you want to have your camera on. It's nice to see friendly faces during this or at any point. And let's get started. Nina Whitney got her start at Carleton College in Minnesota, earning a bachelor's degree in geology. After that, she went to the University of Maine at the Climate Change Institute to pursue a master's in quaternary and climate studies. And then she moved to Iowa State University, where she earned a PhD in geology with a co-major in environmental science, graduating last year. Dr. Nina Whitney is now at Woods Hole Oceanographic Institution, where she holds a NOAA Climate and Global Change postdoctoral fellowship. Nina, thank you so much for being here and showing us how you do some of what you do. And please go ahead and tell us all about it.
Perfect. Thanks, Maddie. Thank you all for being here. And thanks to Pages ECN for the invite. So let's see. So mostly what I'm going to show you today is some lab techniques of preparing shells. But I'll talk a little bit first about, let's see, we'll stop that, try to share my screen. Sorry, there's a lot going on here, but hopefully, how's that look? Good. All right, perfect. So I'll give a sort of a brief background of what my research focuses on, but yeah, mostly I'll just show you how I cut and polish shells, and then a very, very brief primer on how to date these shells. So I work mostly with Arctica icelandica, ocean quahogs. So these guys are the longest living non-colonial animal that we know of in the world. There are certainly some animals that are thought to live longer, including the Greenland shark, perhaps. But fortunately, these guys grow their shells in annual increments. So we have a very good understanding of how long they live as opposed to some other animals. So they can live up to 500 years. The longest living quahog ever found, ocean quahog, was 507 years old. So of course that means that they provide us very long records of climate because we can get some understanding of ocean conditions from their shell, both the width of their increments. So as I said, they grow their shell in annual increments, and also the geochemistry that's within the shell in each of these increments. So I'm an isotope geochemist. I look at the oxygen isotopes to get at water temperature through time. I also look at nitrogen isotopes and radiocarbon isotopes. Those tell us something about ocean circulation changes through time. So that's sort of the broad overview of what I do. I mostly concentrate in the Western North Atlantic region. My PhD was on the Gulf of Maine. This region of the ocean is warming faster than a lot of the rest of the ocean. So part of the impetus for my work is to try to understand why and to provide a longer context. As many of you know, The best oceans records that we have go back a little over 100 years, and that's in not all locations. And so clams provide us with centuries more data to understand the changes that we're seeing today. All right. Let's see if I can advance. So when Maddie first asked me to give a presentation to you guys, we thought it would be fun for me to make a video of a cruise that I was supposed to be on this week on the ESS Pursuit, which is a working clamming boat out of, I think, Atlantic City, New Jersey, mostly. And so I was supposed to go out on this to collect clams, and so it would have been a fun video. But about a month ago, I got an email that said that one of the engines on the ESS Pursuit had blown up. I don't really know what that means, but I know that the boat is now in dry dock, and so we did not get to go on this cruise. But hopefully at the end of August, I'll get to go out there and collect some clams. Because I know last time we got together, Dan gave a great presentation on growing clams in Biosphere 2. Mostly, there has been some work certainly with tank experiments with Arctica Icelandica. These clams grow pretty slowly. So most of the clams that we have are coming from the ocean. We dredge them up with scallop dredges. And obviously those are what provide us with the centuries long records of climate data. I did just want to mention this cruise because it is supposed to go out at the end of August. And it's led by Roger Mann at Virginia Institute of Marine Science, as well as Eric Powell down at the University of Southern Mississippi. Those guys are mostly fisheries ecologists, so they're interested in the ocean quahog fishery, as well as the Atlantic surf clam fishery. So the sort of primary purpose of this cruise is to follow this track. And I know there's a lot of data points here, but the blue line is supposed to be the cruise track. And those crosses are where we're hoping to sample. The issue with these two fisheries is that because of warming waters, the surf clams are moving offshore. And that means they're encroaching on ocean quahog territory because the ocean quahogs live slightly deeper waters, slightly colder temperatures offshore. Surf clams live onshore. As the surf clams move offshore, they're mixing, essentially creating an ecotone where these two habitats, both the surf clams and the Arctica Icelandica, living in the same location. Because the Arctica Icelandica don't seem to be moving yet. It's thought that perhaps they can bury down deeper in the sediment and weather out essentially some of the more extreme temperatures that they're currently experiencing. So that we haven't really seen a shift in their habitat range on the western North Atlantic coast, at least recently. But the surf clams are shifting, and this is a big issue for the fisheries because generally, or at least this is my understanding, I'm certainly not a fisheries person, but my understanding is that when clammers go out, they're either looking for surf clams or ocean quahogs. They don't have permits to bring both back to port, so they have to sort them out at sea. That takes a lot of time, and time equals money. So the main purpose of this cruise is to try to figure out how much of an issue this is. All these little colored dots that you see are places that have been reported that there is an overlap in these two habitats. So in August, we will go back and forth across the shelf to try to systematically document how much of a problem this is and whether the fishery needs to change its regulations and its policies in order to deal with this issue. That's sort of an aside, but I thought I'd mention because it is interesting. Why I'm on the cruise is because they're going to be pulling up hundreds of Arctica Icelandica shells. And so Roger and Eric have been very kind to me and have we've worked out a collaboration, which has been great since they're mostly interested in growth rate and things like that. And then they send clams to me and I can look at the isotope geochemistry and cross-state them, which I'll show you. So we're using these shells that are collected for multiple purposes. And in fact, the shells that I'll show you today came from a site off of Long Island, which you can see up here in the right-hand corner of this map. that were also collected by Roger and Eric in 2017. So let's see if I can advance again. Because you guys didn't get to see a fun video of a clam cruise, I thought I would share a quick video of a different cruise that I was on recently. So this one was in the beginning of April. It was on the RV Endeavor, which is a UNOS vessel. It's the University of Rhode Island boat. And we went out into the Gulf of Maine, primarily to test the instrument that Dr. Adam Shabas, who's here at HUI, developed that looks at sort of testing saturation state of different organisms in situ. So that was the primary purpose, but I was invited along primarily to help with collecting samples and so forth, but also given the opportunity to collect my own samples. So this is sort of a side research that I do. But I look at isotopes in modern water, so mostly oxygen isotopes, and then I've also looked at nitrogen isotopes in dissolved nitrate to give us some baselines for the current isotopic conditions, because of course the clams are getting their isotopes from the seawater. So for example, and I'm sure many of you are aware of this, but the oxygen isotopes that we use in the clams to look at temperature through time, they're essentially taking the oxygen out of the water and fractionating it based on temperature. But that baseline, what they're starting with, is the oxygen isotope in the water itself. Oxygen isotopes in water vary with salinity. They co-vary together. And so we don't have really many records of oxygen isotopes in the water through time. We have more records of oxygen isotopes, of salinity. And so some of my work research has focused on understanding the relationship between salinity and oxygen isotopes in different environments, to try to get at oxygen isotopes of water based on salinity records. So that's sort of why I look at isotopes in the water column. So I'll show this quick video. Let's see. I'm hoping that it'll be okay over Zoom. I will put a link to the YouTube, or I think, yeah, I think I can send this to everyone. So this is the link for the YouTube in case this doesn't work or if it sounds bad. And I apologize, the volume is a little iffy on this, so I'll try to change it as it goes, but hopefully it'll work. It's only 4 minutes, so if it's really painful, just mute it for 4 minutes and you can watch the video later. But Maddie, tell me if it's really not working out. So this is just a little overview of what the cruise was. You'll see some time-lapse images of us collecting water samples from the CTD, which is this big rosette thing in the background here. Basically, for those of you who aren't familiar, you can trigger this to close at different depths. These are Niskin bottles that are on this CTD rosette. So we're collecting waters from different depths all throughout the water column. We went to 2 locations, 3 locations in the Gulf of Maine. The maximum depth is at the locations we were at was about 290 meters. So not super deep, but still very interesting temperature and salinity profiles that we found there. All right, so I will attempt to play this. That was just a little joke. We had to get tested twice in quarantine, obviously, since this was all in time of coronavirus. So it was a lot of going to Convenient MD. This is the Cape Cod Canal.
Josh. I'm an assistant scientist at UI in the chemistry department, and I study calcium carbonate cycling and organisms that grow their shells in the ocean and how they kind of help the ocean take up and sequester carbon. This cruise was funded as part of a project that I have looking at the solubility of calcium carbonate minerals and how that might play into how much carbon, essentially how much buffering capacity the ocean has, and therefore how much carbon the ocean can actually store on various time scales. I had this instrument that was designed to kind of test this solubility parameter experimentally. So we were testing that out here, and then we had a whole bunch of other time over this 5 days. So we tried to just put together as many people as we could to sample a whole bunch of stuff in the water column and particles. So calcium carbonate concentrations, organic carbon concentrations, plankton characterization, flux estimates based on weird thorium isotopes, proteins and metatranscriptomics and all sorts of good stuff. So yeah, that's what we've been doing out here.
Sorry. The reason they keep showing those water jugs is another sort of inside joke, that it took so long to filter all of this water that was for inorganic and organic carbon, just because it was so full of all sorts of living things. It was April, you know, the phytoplankton bloom was coming. There was a lot of stuff in the water. This is a phytoplankton tow that they're doing there with the bongo nets. All right. So that was just a brief introduction to, or sort of overview of what a research cruise can look like for those of you who haven't been on one. It was about 6 days, I think, and this one we didn't have any shifts. Sometimes you have shifts, so you have 4-hour shifts or 6-hour shifts. This one we were just on all the time, so there was very little sleeping, but otherwise it was really great. So the boat operates 24/7. You're just sort of up when there's sampling going on, and there was a lot of sampling going on, but it's a lot of fun, and we had really great weather, so that was good too. But no clams were collected because this is not a clam cruise, but hopefully in August I'll be able to go out and get some clams. All right, so moving on, I thought I'd get a super brief overview of the clams that I'm gonna show you today, why I'm looking at them. So this is June temperatures averaged over some period of time that I'm forgetting, I think 30 years, probably not that long. Anyways, June temperatures, the bottom or the minimum temperature for the region, which usually is close to bottom temperatures in much of this region. And it's showing the cold pool. So this is a parcel of water that forms on the mid-Atlantic Bight region. It's left over and sort of trapped below the thermocline from last winter's cold water at the surface. And it's a really important feature for this region in terms of several fisheries. So because there's sort of unusually cold water here, there are certain fisheries that rely on this cold water to spawn. It limits the habitat. So for example, surf clams live inside of this cold pool region. And then it allows Arctica Icelandica to live there because of the cooler waters. They're certainly close to their southern extent. Down here, the maximum southern extent of Arctica Icelandica are Cape Hatteras, but this is pretty warm temperatures for them. So the fact that this cold pool exists for a good chunk of the year is important for those as well. However, there's some suggesting that this cold pool is shrinking. That's part of the reason, right, that the surf clams are moving offshore. But we have very little bottom water temperature data from this region, like much of the ocean. And so the goal with this project is to reconstruct temperatures at the bottom. Generally cold pool temperatures. Arctica Icelandica are living at the bottom, obviously, so they're recording temperatures at the bottom. We have clams here that go back to about 1850, so I don't plan for this to be a super long reconstruction in paleo terms. This is very short, but it's certainly going to be longer than any instrumental records we have. There's not a lot of fossil shells that you find in this area, so all of the shells that I'm working with and I'll show you today are all live caught. So the oldest ones are about 150 years old, and they don't seem to be any older than that in this region. Certainly farther north you go, the longer they tend to live. And I think in part because all of these shells were caught by fisheries ecologists, they're not as interested in the dead shells. So there aren't any, available archives of dead shells. Hopefully when I get out there in August, we might pull up some fossil shells. And as I'll show you the dating, those are important because then you can start to extend those records that you have. So that 150 years back through time. But right now we have 150-year records. And sort of my goal with this project is to create records from 3 different regions, at least, as you can see here in the stars, of that sort of in the core of the cold pool and then on the edges of the cold pool, to see if I can reconstruct changes in the cold pool through time and see if what appears to be a shrinking of the cold pool recently has happened before, if it's unusual or if it's unprecedented, and at least the last 150 years, which I totally acknowledge is not a super long time. So that's sort of the very basic overview of what I'm hoping to do with this project. So now I'll talk a little bit about some lab processes that I have been doing here at Hui, and I'll show you a little bit of those behind me. So here I am cutting shells with a rock saw, which is a really terrible idea. But it's the only thing I could find at Hui. I'm sure somewhere. So usually what I would use is a diamond bandsaw, like a griffin saw, but I can't find one here at Hui, and I don't really have very much research funds, so we ended up with the rock saw, which was a little scary. These shells are, fairly fragile, but fortunately, most of them didn't break. The ones that did break broke in convenient locations, so I just sort of stuck them back together with some epoxy, and they're fine. By convenient, I mean like regions where we aren't really going to sample them for isotopes and where it's easy to measure around the break. So anyways, that was a little exciting. Don't do this at home, folks. It's not a good idea, but we got it done, and it was certainly a lot faster than cutting them with a bandsaw. This was like super fast. So I think I might stop sharing my screen so you guys can see some props that I brought with me. But this picture on the right, so you can see the shell. Basically what we want to do is we try to cut a strip about a couple of centimeters on either side of the axis of maximum growth, which is essentially just the longest or where they grow the most. Because when we're milling for oxygen isotopes, those increments, especially if it's a long-lived clam, they grow every year, but they grow slower as they get older. So in order to get sort of the widest increment that if possible, you try to cut it along the axis of maximum growth. And this picture is not great because I didn't do a great job. The rock saw is difficult to actually line it up on either side of the axis of maximum growth, but you get the idea. You essentially want to strip kind of where it's growing the most. And then, so that's that. So that's I cut this with a rock saw. Then you block them in epoxy, which is the picture down here, all the different shells in epoxy. I'll show you that. I have an epoxy mold, which generally we use, but as you can see here, there's a lot of different molds, and that's because epoxy molds are expensive. So I try to use recyclables, which some were better than others. I highly recommend ice cream cartons for epoxy molds. Those worked quite well. Some of the other ones, milk cartons, do not work great. This is like a cider carton, also not great. So anyways, adventures in trying to like figure things out where nobody really does clam work at Hui. They used to, but mostly now it's coral. There's a lot of coral people here, some otolith people, but no clam people. So trying to find the appropriate equipment without funds has been exciting. So let me see, I'll stop sharing and then I'll show you. So again, this is a strip of a clam cut with a rock saw. And so then what we do is take, so this is an actual epoxy mold. It's kind of a big one. Generally, I would use a smaller one, but this is the one I have. So you first pour first layer of epoxy in there, and then you put the shell down like so. If I can show you without covering it up with my hand. And then you put a label down. So part of the reason we block these with an epoxy is to sort of make them less fragile so that we can easily work with them. It's also to allow us a polishing surface so that we can polish them so they're super, super polished, which allows us to look at the increments under a microscope. And I guess the third reason would be to put labels into the epoxy with them so that we don't get confused about which cell is which, because I mean, they all are individuals, they have their own quirks, but they generally look the same. So you don't want to screw them up, mix them up. So we do that, you pour another layer of epoxy, let that dry. The epoxy these days takes about 6 hours to dry, which is super fast and also a little troubling because it's exothermic. It like heats up because it's drying so fast. And that makes me nervous because I don't know, I don't want heat near my clams. So I might have to look for another epoxy, but it dries pretty fast. Then you put another label in because what I'll show you in a second is you're going to cut these epoxy blocks in half. Then you fill it up. So what you have after you do all that is an epoxy block like this. And it's got 2 labels in it. You probably can't really see, but we're going to cut this in half essentially. And so then we'll have 2 mirror images and they'll both have a label. Sometimes what you'll do is etch one with hydrochloric acid, and that sort of essentially loosens the top layer of the shell. And then you put down acetone and acetate paper and peel off the top part. And that gives you a slide that you can shine light through and look at under a microscope. And that is sometimes how we use to date the shells. One of the big things that I learned in my PhD is that I'm really terrible of making acetate slides I'd make 20 and maybe like 3 were good. And it's time consuming. And if they're not good, you have to then go back and usually repolish the shell again, which is, an hour or more of work, then etch it again, and then hope that your acetate peel is better. So I've tried to stray away from acetate peels, so I don't have any to show you today, and just date it on the block itself. Because I find that generally, depending on the shell, some shells you really need that slide, but most shells, if you polish it long enough, you can date it on the block. So that's good. All right, I have to stop talking because I just yapping away. So I will show you guys a couple of things. So first of all, this is the Isoma low speed saw that we use to cut those epoxy blocks. I've already sort of set up a shell here, so I'll just let it run for a second so you guys can see. But basically, it's a diamond wafering blade. There's water that's sitting in the chamber to cool the blade and also clean it, and plus there's some sponges down there. You can see, hopefully, that essentially that blade is lined up with the middle of this shell. So again, we're cutting the shell again in half. So we're taking this piece that I've cut and cutting it in half. So we have two mirror images. Oh, I was gonna say, so if you etch one of the shells, the other one you can use for isotopes, because you don't want to do isotopes on the shell that you've put in a hydrochloric acid. So that's sort of the benefit of having two mirror images, is you can etch one and then the other one save for the isotopes, where we micromill within the shell. Since I'm not doing or trying not to do acetate peels, generally, I just will polish one side and I'll date it and mail it for oxygen isotopes on the same side. So that is my goal, at least. We'll see how it goes. So anyways, I will just start running this. I've basically put the shell in a check. Can I show you? So it's just a thing that holds the shell onto this saw. So I think if I put this down, so essentially what I've done, this is an arm that you can move up and down. You secure this chuck to the arm. Sometimes. Damn it. There we go. You can move this back and forth so you can change where the blade is going to hit the shell. But I've already set it up so that it's going to cut in the middle of that shell, theoretically. And then you start it. Goes pretty slowly. You have to put weights on top of it so that it sort of doesn't bounce on there and it holds it down. And then it just goes. So it'll take a good, depending on the shell, sometimes they'll take 15 minutes, sometimes they'll take an hour or more. You have to often change the angle quite a bit because it'll sometimes get down to, basically it'll hit this part. And so then you have to change it around to cut every side of the shell. So it takes a while, but that's how we cut the shells. And then the resulting blocks are here. So these are two different shells. I've already put away their mirror images, but you get the idea that they would they would essentially go together like this. So you cut it in half and then you get two mirror images and you polish now both of these because they'll have saw marks on them and various other things. And I'll show you the polishing. So we start, this is the polisher. We do 120 grit, 400, 600, 800, and 1200. So progressively getting finer grit. It runs like that at about 300 RPM. You can see that it used to be plumbed for water, but now it is not. And so we've just run a hose from the sink and zip tied it onto here because you need water so it'll be cooled down and also to sort of keep cleaning the polishing surface. That water drains into here, and this is the trap that I've hacked together to try to collect some of that epoxy because otherwise it would really plug up the drain. So I'll just briefly show you. I know I'm conscious of the time, so I'm not going to spend very much time at all. But this is 600 grit right here. So I've been sort of, these shells that I'm about to polish have been polished at 120 and 400, and now I'm polishing them at 600. So stay there. Start the water. It's like very finicky. Start the water, and then you just take your shell. And then you stand here for hours watch a Netflix movie. I've been trying to watch documentaries to at least feel like I'm, you know, improving my brain somehow. But it's a long process. So I think Maddie was the one that taught me basically what we're looking for, and you can sort of see it, is that when you hold them this way, they'll get smiley faces of that sort of indicate the epoxy just sort of aligns in a smiley face because it's spinning like this. So you'll get this pattern on here. And so once it's all the same pattern of smiley face polished surface, you know that it's fully polished on that side. And then you can move on to the other grit. You turn it around and you look for the frowny faces. So then the sort of the surface will all appear to have subtle polish lines in the other direction. And so once they're all frowny faces, you know that it's completely polished on that next grit and you can move on to the next grit. So that's the trick to that. But all right. So I just have a couple more things to show you and then I'll be happy to take questions. But I thought I would demonstrate quickly dating of these shells. So let me share my screen again. All right. So this is actually an acetate peel like I was telling you about. Well, obviously this is an image of a shell and then this is an acetate peel. I think from Maddie, so thanks Maddie for this image. But it shows you that these shells, they grow their increments in two locations. So at the hinge area, which is down here, and it's up here, hopefully you can see my cursor, but it's this top region in the A plot. And then also along their entire margin of the shell. So all along the back, they grow in annual increments as well. And so we can, generally you date or measure those increments in both the hinge and the margin, compare them first to make sure you haven't made any errors because they should match exactly. Once you've, they're not the same width, but the same pattern. So you have to detrend them both because they grow faster when they're younger and also to take into account differences between the margin and the hinge. So generally we put negative exponential growth curves on them and detrend them that way. But once you do that, the margin and the hinge should match up like they do in this image here. And then you can compare them to say other shells dated in the region. And this is how you work your way back in time because you have sort of live caught shells. You can compare live caught shells with other live caught shells to create what's called a master chronology and check to see if you've had any errors because they should all match in terms of the pattern of thick to thin increments because all of the shells in a given area have experienced the same environmental conditions, which impacts how fast they grow in a year. And then you can also work your way back in time, assuming the fossil shells you have overlap in time with the live caught shells to some extent. So that's how we date them. I'll just briefly show you that, and I'm going to show you how I would do it in the margin first, and then I'll show you an image of the margin, and then I'll show you an image of the hinge. So let me try to get out of here and pull up. All right, so this is the margin, and I'm doing this in image J. Let me see if I can make it any bigger. I guess that's as far as we can get, so hopefully it looks okay to you guys. Essentially what I would do is find the image J toolbar, which is hidden somewhere. Here it is. And you have to be careful because obviously these are varying in length as you go. So this is the younger part of the shell up to the upper right. The older part is down to the lower left. But so you always want to measure in the same part of the increment. So I always do it like right before it flares out. So you make a measurement that is perpendicular to the growth lines. So something like that. I'm sort of doing this fast, so not being super specific. And then you do that and it will record somewhere the length of the shell in millimeters. Now, I have not actually calibrated this image in particular, so that length is probably off, but how I would do that. So this is from, we've imaged it using a microscope and a camera attached to a microscope. And then you can go to set scale and, oh, sorry, I have to first draw a line along this scale. So this scale is from the microscope, and the microscope software, not from ImageJ, has no idea how big this cell is. So first, you draw a line along that shell, you do set scale, you tell it that that's 1 millimeter, and then you've calibrated your shell. All right, now that we've done that, we will go back, measure this increment again so we get the correct length. And then keep going down the shell. So I picked images that were pretty clear. Dating is definitely tricky because the shells like to randomly stop growing in the middle of the year. Maybe it was too warm for them. And so it can be difficult to determine. For example, in this one, those ones to me are pretty clear, but this one could be a really small increment here, or it could just be what we call a check. It was just, you know, sort of, it stopped growing for a period of time, but it's not an annual increment. So that's why cross dating can be so powerful, because I'll have to make a judgment call on whether I think this is an increment or not, but then I can go back, check with the hinge. If the hinge doesn't have a small increment here, it's probably not an increment. If they both do, because they both, you know, they both suggest that it's an annual increment, you can then go and look at other shells to see if all of the shells have that same increment. All right, so, and also this is a more clear increment to me. So I would actually redo that and suggest that this is maybe a tiny increment where the shell didn't grow much during that period. I'll quickly show you the hinge, and it looks quite different. There is a bit of a strange crack in this particular shell, so ignore that. But again, we'd have to calibrate it in terms of time. I won't do that, but it is a different magnification. This is 20x, the other one was 10x. So I'd have to calibrate it. But again, you can go down here, trying to stay in roughly the same spot. Oh, sorry. And it'll, again, you're getting the measurements over here. So yeah, so that's generally how we date the shells. And again, I'm going pretty fast, so I'd take a little more time to really look and make sure that you're going right to the edge of that black line every time. Oops. Oh. And let's see. This one looks a little fuzzy, but then you can look on the edges and it's pretty clear there. So I would say that's an increment. This is an increment. This one is a pretty small increment, but it looks pretty convincing to me. So that's, yep, that's a very quick intro to cross dating. I'll show you. You can, oh, it might have timed out. this may take too long, but essentially the next step you would do is then put it into some sort of, there's Matlab Scripts. This is a script, sort of an R script developed by David Reynolds and Brian Black and others, that essentially allows you to compare your increment widths. And I think it's probably going to take too long because I had it set up, but I guess it timed out. So it'll show you essentially where two shells are correlated, or where your master chronology in another shell is correlated, and where there are gaps in that correlation, so it allows you to figure out what errors you've made in your dating. But it's going to take quite a while, so I might stop sharing and take any questions. And I can certainly in a couple minutes show this to you if anybody's actually interested in seeing it. So yeah, thanks for your attention. Sorry that was a bit long, but I'm happy certainly to take any questions that you guys have. Oh, and I see some in the chat. Are these questions?
Oh, they were commentary. I was just saying it's so relatable. Some of the things you brought up about the acetate peels and ImageJ, and it was funny seeing, you know, the things that we do that are the same and the things that we do that are different. And I picked up some good tips watching your walkthrough. So that would bring my first question. When you got started, first of all, this is a great walkthrough for anyone trying to get started with this clam stuff, because a lot of us learn this by trial and error and repeated failure until we find the right approach. How did you end up, you know, coming up with this approach and honing it through time?
Yeah, that's a good question. I would say certainly Maddie taught me a lot, because we went to the same PhD program, and so she was a couple years ahead of me. So she taught me a lot that I know, and then I taught the next students coming in. But yeah, I would say you're exactly right, Dan. It's a lot of trial and error. For example, the fact that I don't use acetate peels was because I was getting so frustrated. And then I had to date some shells that didn't have acetate peels. And so I thought, okay, well, maybe I will just try to date them on the shell itself and not use an acetate peel, because it's going to take me a while to make acetate peels, and realize that, oh yeah, it wasn't so bad. It was actually pretty easy to date them on the block itself, just with reflected light. So yeah, just a lot of trial and error. And I think I didn't really go to any workshops. It was mostly just learning from Maddie and others in the lab and my advisor, Al Wanamaker, and that kind of thing.
Yeah, I'll just add,
Great.
It goes up the chain because I learned from people who were there before me, and they and me as well actually got the chance to attend the Dendroecological Field Week, which is, it's easy to look up, but that's obviously for tree rings. And they actually talk about shell growth there too sometimes, but they actually teach you how to process samples and cross-date annually banded materials at that workshop, summer workshop. Look it up, Dendroecological Field Week.
Yeah, I guess I lied. I did go to that one, so forget about that. That is a great workshop. It's exhausting, and there's so much that gets done in a very short amount of time. And yeah, I think you're exactly right, Maddie. There is a shell component to that. I did the tree component, which was actually really interesting. I already knew how to cross it at that point. It was interesting to see the similarities and differences between dendrochronology and sclerochronology. We certainly use the same techniques in terms of cross-dating, but it's just interesting. Things like trees can just have missing years. They just don't grow that year, and there's no evidence of growth. Shells don't, thankfully, have missing years. I mean, you always are going to have a year during that time, at least all of the shells that I've worked with. But sort of some harder things, like in trees, you can actually look at cells and physically see when the cells are closer together and identify increments that way. And shells, there aren't individual cells that you're looking at under a microscope. And so it's just sort of the dark line that you're hoping is actually an annual increment. So yeah, it was really interesting to see the differences between those two.
Great. And I hope that this video, because we'll be hosting on our website, will be something students can glom onto, find in a YouTube as a YouTube tutorial. What other questions do people have?
Nina, I think you did a good job at showing how really tedious some of the processing can be. Like it's really a lot, like it takes a lot to build these shell growth records, which is probably why there's really not that many of them compared to other types of proxies, which are also in their own way expensive and tedious and whatever. But what is your favorite and least favorite tedious part of preparing shells?
I really like the dating part. It is a lot of looking at lines for literally all day, and it's just line after line after line. But I find it, I don't know, it's kind of a game, and it's fun to run it through the script and then figure out whether you're right on or if it's like totally off and what happened. The polish, I really don't like polishing. It's so I mean, you can spend an hour or two on the same shell and it can seem like you're making no progress at all. And then you sort of zone off and you're watching Netflix or something and you look at your shell and you realize that you have two polished planes, which creates like you can't actually feel it, but you can see a line running straight through your shell. And then you have to go back to like a coarser grit and repolish it because you don't want that it's terrible. So yeah, the polishing is definitely my least favorite. I mean, it is nice. It's actually been a nice break recently to zone because it doesn't take any mental effort. So you zone out and you can think about other things. Because I've been trying to write a paper, so that's been a nice break. But it's very long, and then you like polish off your fingertips too, and you start to bleed on like every single fingertip because you haven't paid attention and you polish them all off, so that can be problematic too.
Yes, I actually also had a question. Thank you so much for giving us this deep insight. It's really interesting, and I have to admit, I'm quite fascinated by shells now. And coming from lake sediments as background. I wondered for the dating, you showed us how to draw lines in ImageJ, etc. And I've seen that obviously the layers are differently width or have different thickness. And how do you determine where to measure the increment? You know, is it? I know there is a systematic behind it, but I couldn't get it yet.
Yeah, sorry, I think rushing through it.
No, that's me. No, it was fantastic. Sorry.
So it's a good question. So the hinge, generally, and it wasn't, they're narrow on the sides and then they come down and it's thickest sort of at the base of the U, I guess, because they sort of make a U like this. So here it's the thickest part. So on the hinge, I try to measure at that thickest part every time.
Okay.
So just to be consistent and follow, sometimes it'll change the direction of growth, but if you always sort of measure at the very base of that U shape that it's making, then you can sort of be consistent. For the margin, it's a little harder, but I was saying they sort of like, they come up and then they flare out right at the end. That flare is not really representative of the growth. So I try to measure at the maximum right before the flare, because the flare does tricky, funky things. I don't think it's really related to its actual necessarily growth. So the maximum, so the thickest it can be without measuring the flare. So right before it sort of goes out, that's where I measure it every time.
Wow, nice, thanks. And also for the dating, usually for lake sediments, if you work with warped sediments, so annually laminated sediments, and also counter layers, we have at least 3 different people doing the counting independently to have sort of a mixed uncertainty calculation. Is there also a similar approach, or depends it in the end on one count, or?
Yeah, that's a good question. Some people do it that way. We don't. So it's, because we're cross-dating, that's sort of our check on the uncertainty. And it's all done by the same person. Well, not necessarily. I mean, I certainly have contributed to other chronologies. Sorry, my light's gone off because I've stopped moving and the motion sense, there we go. So we do not like have 3 people date the same sediments because we do the cross dating. So our sort of check on whether we've made errors is, first of all, do the hinge and the margin match? And then if so, does it match with the rest of the shells that have lived in this population for this time period? But I do know, so these shells actually originally came from VIMS, the fishery ecology people, and they do not cross date their shells, but they do have 3 different people date the shells. So it's just different approaches, but because we're cross dating these shells, it's usually just one or maybe, multiple people working on the same chronology.
Nice. That's awesome. Cool. Thanks so much.
Yeah, of course. Thank you.
Okay.
Yeah, Nina.
Yeah, hi.
Yeah, I'm Onema.
Nice to meet you.
Yeah, nice to meet you too. Yeah, I mean, thanks for the wonderful presentation. And I saw the dimension you were trying to talk in relation to that's a shell of, what you're working on and all that.
Yeah.
But my question comes in this manner. Did you try comparing your information with some other organisms that their shells also dissolve very fast in the ocean, like pteropods, that's the mollusks and all that?
I've never done that. No, compared my current shells. You mean like from sediment cores or?
Yes,
Like,
yes, from the sea. Yeah.
Yeah, it's certainly something like with this project I've just started, so I don't actually have any data from this project yet. But certainly something to look into would be to compare these records to sediment core records from the region. There's definitely work and certainly people at WHOI that have done that. I have with my PhD work from the Gulf of Maine, I've looked at some records of sediment cores from sort of more northern locations. So it's certainly, I think where this science is heading is a lot of the work over the last several decades has been to develop these techniques. But now we're getting pretty confident in our techniques, and now we're starting to create climate records that we can then compare to other records in the region. So yeah, so that's definitely exciting step forward, I think.
Yeah, okay, that's okay. So invariably, you're looking at more of the climate change of the setting. Are you considering the ocean acidification in terms of the dissolution of the shelf and all that?
Yeah, good question. So I don't personally work much with ocean acidification, although I am sort of dabbling in looking at boron isotopes, which you can get pH from, theoretically at least, from bivalves. And so sort of a future goal of mine is to look at boron isotopes in scallop shells, perhaps. That's sort of something we're trying to figure out right now. In terms of the dissolution of the shells, at least for Arctica Icelandica, we're still trying to figure out how ocean acidification is going to or is affecting Arctica Icelandica shells. There is some recent studies that suggest that because they can regulate the pH in the extrapaleal fluid, which is basically where they're creating these shells, that region, they may be less impacted by ocean acidification because they, to some extent, can create their own pH. So their shells may be less susceptible to being dissolved. But I'm certainly not an expert in this. But that's sort of where the science is right now. We're not quite sure how and when and if ocean acidification is going to affect Arctica Icelandica. I don't think there's really any data that suggests that it is currently affecting these clams at this point.
Okay. Yeah, thank you very much.
Yeah, of course.
Yeah.
Thank you.
Those are great questions, everyone. Thank you for attending. You can probably hang around for a few more minutes, but if there's any remaining questions, now's your chance. Otherwise,
Hello.
okay, there we go.
Hi, how are you?
Good,
Nice,
how are you?
nice presentation. Thanks for showing us. I was wondering how time consuming is the work for geochemical analysis, microdrilling, or something like that. If you do that, if you make that kind of work, if you can tell us about it, please.
Sorry, can you repeat how time consuming is the what?
The microdrilling, I don't know, for isotope analysis, you use what kind of
Yeah. Oh,
spectrometer you
yes.
use, or-
Micromilling. Yeah. So the micromilling, and sorry, I don't have one here to show you. It's also very time consuming. I feel like Maddie is a much faster miller than I am, but generally I can mill in an 8-hour day, like 11 increments. So it's very like slow going. But yeah, so basically the way it works is that you control the micromill, which is attached to a microscope on a computer. You tell it exactly where to mill. It's an 80 micron diameter bit, and then it will mill out that section of the shell. So some people do seasonal work. I've always done just annual work. So you want to get essentially the whole annual increment, get all of the carbonate powder from that one increment. And so, yeah, it's pretty time consuming. You know, if you want to get 200 years of data, it'll take many days, 10, 20 days to get that much data. Because you set it up, you let it run, it maybe takes 10 minutes, sort of a normal increment, but sometimes if it's really thin, you have to go pretty deep to get enough powder, and so that can take a lot longer. So the mill can be running for 30 minutes or more, and you're sort of just sitting there waiting for it to do its thing. What else about micromilling? Yeah, I think that's sort of the basics of it. I feel like I was gonna say something else. Oh, the other thing is that generally, if you're trying to get annually resolved data, the shells grow slower as they get older. So it's really hard in a 200+ year shell to mill the last, like the older increments, because they're really, really narrow. So generally I've found it's fairly easy to mill the first 20 to 30 years of the clam's life, but then you have to switch to a different clam and hopefully you have enough clams that overlap where you have some really old ones, and so you can take the 30 years, their first 30 years of life, and then some younger ones, so you can take their first 30 years of life.
So
yeah.
for that is very helpful, the cross dating for that kind of.
Yes, exactly. Yeah, you want shells. And so even with the shells I'm working with, even though they were all live caught, some of them are 150 years old. So I know I'm going to be able to get sort of the 1850s to 1880s or 90s. And some of them were born in the 1970s. And that's really important because I know I'll be able to get both sort of the 1800s and also the much more recent time, which of course we need to compare to the instrumental record. So yeah, so it's important to have shells of different ages within your cross-dated master chronology.
Okay, thank you very much.
Yeah, thank you.
Okay, well, I see a lot of names on here that I recognize who are very familiar with Pages ECN, but if you're not, I'll just share this final plug. So make sure you're signed up for the mailing list, and you have a Pages profile and everything, so you can be as involved as you can in our early career network. We have a really good time and do a lot of really nice networking things. So again, Nina, thank you so much. We're going to have another event, I think, next month, so stay tuned for an announcement. And If no one else has anything to add, you can give Nina a little emoji or a round of applause or a thank you. And I see there's some compliments in the chat box as well. So thanks again, everyone. And have a
Awesome.
lovely
Thank you
week.
guys so much for coming.